Hi, my name is Eva Chin. I established a lab where we use animal models of muscle diseases such as muscular dystrophy, mice such as the ones shown up on the wall. And then specifically we started characterizing defects in their muscle function. And particularly I'm interested in the calcium signals in muscle. So I isolate muscles, I isolate the muscle fibers, and then we look at the muscle fiber function, and we place them in this chamber, which we sit on an imaging microscope. And by characterizing the defects in muscle function in these single muscle fibers, we found defects in a number of muscle diseases, muscular dystrophy, as well as we started looking at an animal model of ALS, or Lou Gehrig's disease. So after the initial characterization on the physiological changes, we then went and looked at some of the protein level changes. We also do a lot of molecular biology, looking at changes in protein level, changes in gene expression, and we've identified several protein factors and several signaling pathways that are altered both in muscular dystrophy as well as in ALS. Most significantly, the invention is based on our work on ALS, or Lou Gehrig's disease. It's a severe neurodegenerative disease where the muscle starts to atrophy and dies off. Patients become weak. They have problems walking. They become wheelchair bound. They develop problems speaking and swallowing. And eventually, they die of respiratory failure. With the invention that I've disclosed, the hope is to take our knowledge about these protein defects in ALS and to translate them into a protein test. So our plan is to take the proteins that we have identified and develop an assay that can be run in the standard diagnostic lab, something like this in a 96-well format, so that they can get information on all these different proteins that we have identified. And through combining that with clinical data, we hope to develop a method for both earlier diagnosis of ALS, so patients can determine they have the disease earlier, which enables earlier treatment and hopefully better outcome, better quality of life. We can also use this in clinical trials. When they study the effect of new drugs, it is helpful to have some type of assay system. We're moving forward and trying to take what we've learned from characterizing muscle function into some form of a translatable assay that clinicians can use to help in diagnosis and in monitoring their patients. So again, our hope is to take this into the clinical realm and really develop a product that will have impact for patients, that will help them in their quality of life, and of course in finding a new drug through clinical trials. ALS right now only has one drug that's approved for it, and it only extends life by 2 to 3 months. Certainly what we've learned is that clinical trials means hope to these patients.
My name is Frederick Kotchik. I'm a senior research scientist at the Department of Chemistry and Biochemistry. I have been working with carotenoids for almost 30 years. Carotenoids are a group of naturally occurring compounds that are abundant in foods, in flowers, plants, and animals. We absorb about 12 to 15 of these, and these carotenoids are found in our circulatory system, in organs and tissues, and have been shown to prevent a number of chronic diseases, such as cancer, cardiovascular disease, and eye disease, such as macular degeneration and cataract. I realized that there are certain requirements for carotenoids to be absorbed into the human eye, particularly the macula. And because of the structural similarity, I decided to investigate another carotenoid, which is called Betacryptoxanthine. Betacryptoxanthine is a rare carotenoid that is present in our diet. It accumulates similar to lutein in human organs and tissues. And I decided to investigate this carotenoid because there have been several animal studies as well as human intervention trials that have shown beneficial effect of Betacryptoxanthine in the prevention of calcium, bone calcium, and also lowering the blood pressure. And Betacryptoxanthine also accumulates in the human ocular tissues. I have done a lot of work in that area that shows that Betacryptoxanthine, for example, accumulates in the human ciliary body, which is a tissue that is responsible for contraction and expansion of the lens. And when it doesn't function properly, it results in a disease called presbyopia. The total synthesis of betacryptoxanthine requires in excess of 10 synthetic steps and uses very harmful and toxic chemicals. So I decided to come up with a methodology that would take commercially available lutein and converts it directly to betacryptoxanthine. Now, the interesting part about this transformation is the fact that there are basically 4 reactions involved, and I was able to accomplish this transformation in one single process, in one pot reaction, and under very mild condition using basically green chemistry. So by having betacryptoxanthine available in large quantities in very high purity, one can therefore conduct further animal studies and perhaps human intervention trials to investigate the beneficial effect of betacryptoxanthine in lowering the incidence of heart disease, for example, because betacryptoxanthine also is a very efficient anti-inflammatory agent. And also, similar to lutein, perhaps it could be used in studies of macular degeneration.
So my name is Jim Culver. I'm in the Plant Sciences and Landscape Architecture Department, as well as in the Institute for Bioscience and Biotechnology Research. We focus on is trying to incorporate biology into novel nanoforms and structures that are can be used in different devices. We work primarily with a plant virus called tobacco mosaic virus, and one of the things that we've done in the past is incorporate different amino acid residues, different peptides, different structural features onto the virus particle itself in order to impart these particular traits, such as sensor development and electrodes. We developed an in vitro or a bacterial expression system that allows us for the first time to assemble these virus rods in the bacteria themselves without the need for virus replication or any sort of plant process. And it makes it much easier and much simpler to produce these rods that have very unique and varied sorts of structural features added to them without any problems of the virus recombination that might lead to the loss of traits that we're looking for. And from there, we're hoping to incorporate these particular particles into cell delivery devices, electrodes for different sorts of battery operations, as well as sensors displaying various receptor peptides and these sorts of things onto the surface. For the technologies that we're interested in, adding to the VLPs or the virus-like particles that we're producing are novel peptides and residues that we can use to attract or different metal ions onto the surface of this biology and give them unique traits to let them function in certain battery operations. Also, positioning the different residues specifically within the structure of these virus-like particles. We can orient the particles, these long tobacco mosaic virus-shaped, rod-shaped particles, so we can create a forest of these particles, nanoparticles, on a particular surface, and this enhances surface area and thus enhances the function of the device, whether it's an electrode or a sensor in which the virus-like particles are displaying receptor peptides for various, say, pathogens or other sorts of chemicals that we might be interested in looking at. Or for therapeutics, we're very interested in using these VLPs to encapsulate novel nucleic acids that could be used as therapeutics for drug delivery into cells. In particular, within the GI tract is a potential area that we're interested in.